Welcome back to Exploring with the Bio Twins. Today we'll be talking about macromolecules. Get ready for this exploration into the building blocks of life.
When we were younger and watched TV all the time, we noticed there would always be commercials about fat.
They would tell us to stay away from fat.
They would say how bad fat is for you.
They would promote fat-free products.
And they would talk about how fatty foods actually made a person fat.
Fat seemed like this evil creature that would sneak into your body and make you gain a lot of weight.
But what are fats really?
Fats are a type of lipid. Lipids are macromolecules.
Macromolecules are large living molecules that make up all living things, even the foods we eat. There are 4 types of macromolecules:
carbohydrates, lipids, proteins, and nucleic acids.
Macromolecules are considered large because they're made up of small repeating units. These repeating units are known as monomers. Monomers are the building blocks for all macromolecules. Monomers are like individual bricks.
When monomers join together, they form a polymer, which is the macromolecule. Each macromolecule has its own monomer. Carbohydrates are made up of monosaccharides. Lipids are made up of fatty acids. Nucleic acids are made up of nucleotides. And finally, proteins are composed of amino acids. Now on to each individual macromolecule. Each one has its own function and purpose.
Carbohydrates are one of the most well-known macromolecules, often referred to as CARBS. They provide the body with quick energy, and their main function is to be a source of energy. Foods that are carbohydrates are bread, pasta, and sugary desserts. Their monomers are monosaccharides, also known as the simple sugars. This can be glucose, fructose, or galactose. Next, let's talk about lipids. Similar to carbs, lipids provide the body with energy. However, lipids are a source of long-lasting energy and stored fat that isn't depleted as easily as carbs. Lipids are also great for insulation and keeping the body warm. Examples of lipids are butter, oil, and avocados. Unlike every other macromolecule, lipids don't have true monomers, but their building blocks are fatty acids and glycerol. There are 3 types of lipids: triglycerides, phospholipids, and sterols. Triglycerides are composed of a glycerol and 3 fatty acid tails. Most of the lipids that we eat are in the form of triglycerides, such as butter and oil. Phospholipids are composed of 1 phosphate head and 2 fatty acid tails. Phospholipids are what make up the cell membrane, also known as the phospholipid bilayer. The fatty acid tails of a phospholipid can either be saturated or unsaturated. The difference between them is that the saturated fats are saturated with hydrogen atoms and lack double bonds. Saturated fats tend to be solid, while unsaturated fats tend to be in the liquid form. Sterols are the last type of lipid. The most common is cholesterol, which provides structure and permeability to the cell membrane. Now on to the next macromolecule, proteins.
Proteins are made up of amino acids. Amino acids are the monomers of proteins. Amino acids link together to form a chain that will eventually become a protein. Amino acids are composed of 3 distinct groups. There is the carboxyl group, the amine group, and the R side chain. Each of these groups is bonded to the base carbon. The R side chain is what makes each amino acid unique. Amino acids link together to form a chain. This chain is known as a polypeptide. The peptide must go through a series of folding to become a protein. The first in the protein folding series is the primary structure. This is just the polypeptide chain of amino acids. The next in the series is the secondary structure. This is when beta-pleated sheets and alpha helixes begin to form from the amino acid chains. Next in the series is the tertiary structure. This is when 3D structures begin to form as a result of R side chain interactions. Each protein has a different R side chain. The different properties of the different R side chains cause them to interact and create these 3D structures. The last in the series of folding is the quaternary structure. This is the protein consisting of multiple amino acid chains. The last, but definitely not least important macromolecules are nucleic acids. The most known examples of nucleic acids are DNA and RNA. The monomers of nucleic acids are nucleotides. There is a nitrogenous base and a phosphate sugar backbone. First, let's talk about DNA. DNA is the type of nucleic acid that's found in the nucleus of eukaryotic cells. It is double-stranded. DNA has deoxyribose sugar. DNA also has 4 nitrogenous bases. They are adenine, guanine, cytosine, and thymine. These are complementary base pairs, which means that they only pair up with another specific base pair. Adenine pairs with thymine, and guanine pairs with cytosine. DNA is also exclusive to the nucleus of eukaryotic cells. It will not be found anywhere else, except for in the mitochondria, but that's a whole nother story. Next, let's talk about RNA. RNA has the same monomer as DNA, a nucleotide. It has a nitrogenous base with a phosphate sugar backbone. Despite them having similar names and the same type of monomers, DNA and RNA have some distinct differences. RNA is the type of nucleic acid found in prokaryotic and eukaryotic cells. It is single-stranded. RNA has ribose sugar. RNA also has 4 nitrogenous bases: adenine, guanine, cytosine, and uracil. Uracil is replaced for thymine in RNA. That means that it also pairs with adenine. RNA can also be found in the cytoplasm and nucleus of eukaryotic cells. Because prokaryotic cells have no membrane-bound organelles, RNA is also found in the cytoplasm of prokaryotic cells. Well, that's all we have for you today. We hope that you enjoyed learning about macromolecules with the BioTwin. If you're interested in learning more about anything mentioned in the videos, including phospholipids in the cell membrane and protein folding, check out our other videos on those subjects. Until next time, see you later and continue exploring. The BioTwins are out.